this video, we are going to study about the biological molecule that is protein. Under this video or under this topic, we will discuss about what is protein, structure of amino acid, different types of amino acid, peptide, bond, shape of protein, classification of protein, level of structure or organization of protein, and function of protein. So let's start it. Protein name is derived from a Greek word protos, which means the first or the supreme. Protos also mean very important. Protein are extremely complicated in nitrogenous molecule made up of variable number of amino acid residue joined to each other by a specific covalent bond called peptide bond. 20 amino acids which have been found to occur in all protein known as standard amino acids. These are the various definition of proteins. Polypeptide chain of amino acid is simply known as protein. The molecule which yield amino acids upon hydrolysis are called protein. Proteins are natural polymers of amino acids. Amino acids are monomers. When many amino acids are combined with one another, it produces a polypeptide chain, and this polypeptide chain is known as a polymer, which is protein. The number of amino acids in a protein molecule may range from 2 to several thousand. Protein molecule contain nitrogen, carbon, hydrogen, and oxygen. And some protein also contain sulfur. Protein are made up of smaller units called amino acids which are attached to one another in a long chain. There are 20 different types of amino acids that can be combined to make a protein. Amino acids are coded by a combination of 3 DNA building blocks, that is nucleotide, nitrogenous base, pentose sugar, and phosphate bond, determined by the sequence of the gene. The importance of protein was recognized by chemists in the early 19th century, including Swedish chemist John Jacob Berzelius, who in 1838 coined the term protein for the first time, a word derived from the Greek proteios, mean holding first place. What is amino acids? These are the structure of amino acids. Protein is made up of a long chain of amino acids. There are 20 amino acids. The specific order of amino acids determines the structure and function of each protein. Amino acids are organic compounds that combine to form proteins. Amino acids in proteins are the building blocks of life. When proteins are digested or broken down, amino acids are produced. The human body uses amino acids to make protein to help the body and break down the food. Amino acids are a group of organic compounds containing two functional groups. A bifunctional organic compound is known as amino acids. And which type of functional group is present? Amino and carboxyl functional group is present. NH2 is known as the amino and COOH is known as the carboxyl. So a bifunctional organic compound in which the amino group as well as the carboxyl functional group, they are attached to the same carbon atom. Such bifunctional organic compound is known as amino acid. The amino group is basic while carboxylic group is acidic in nature. The amino acids are termed as alpha amino acid. If both the carboxyl and amino groups are attached to the same carbon atom, the alpha carbon atom bind to a side chain represented by R. Apart from the R, remaining structure is similar in all amino acids. So that we can say that these are the constant. R, which is different for each of 20 amino acids found in protein. The amino acid mostly exist in the ionized form in the biological system, mean in the cell. The first amino acid which was discovered is asparagine in 1806. And asparagine is discovered from asparagus plants. Threonine was the last amino acid to be found in the year 1938. All the amino acids have trivial or common name from which they were first isolated. Asparagine was found in asparagus plants and glutamine was found in wheat gluten protein. Tyrosine was first isolated from cheese, and glycine, Greek glykos means sweet, was so named because of the sweet taste. Classification of amino acid based on nutritional, based on polarity, based on their metabolic fate, based on their structure, and two main groups of amino acids. Nutritional classification of amino acids. The amino acid can be divided into two types based on nutrition. Essential or indispensable amino acids and non-essential or dispensable amino acids. The amino acid which cannot be synthesized by the body and therefore need to be supplied through the diet is called essential amino acid. Certain amino acids come under this group. Their example is arginine, valine, threonine, and tryptophan. These are essential amino acid as well as their structure: arginine, lysine, styrene, valine, leucine, isoleucine. Non-essential amino acid. These are those amino acids that the body can synthesize, and approximately 10 essential amino acids meet the biological need. Hence, they need not to be consumed in the diet. Their example is glycine, alanine, serine, etc. These are the example of non-essential amino acids. Classification of amino acids based on polarity. Based on polarity. Non-polar amino acids with aliphatic or groups. These are various types of classification of amino acid based on their polarity. So the amino acid can be divided, 20 different types of amino acid can be divided into 5 groups based on their polarity. We will discuss one by one it. Non-polar amino acid with aliphatic R group. The amino acids, these amino acids are non-polar and also referred to as hydrophobic. Hydro mean water, phobic mean fearing, water hating. So they have no charge on the R group. The amino acid included in these groups are glycine, alanine, leucine, isoleucine, valine, and methionine. Non-polar amino acids with aromatic R group. As the name that ring-shaped amino acids are known as non-polar amino acids having R group, aromatic R group. Their aromatic side chains are relatively non-polar, hydrophobic, cannot dissolve easily in the water. All can participate in hydrophobic interaction. The amino acids included in these groups are phenylalanine, tryptophan, and tyrosine. Polar amino acids with no charge on R group. These are the non-polar aliphatic R group and these are aromatic R group. The amino acids carry no charge on R group and are soluble in water are more hydrophilic. Hydrophilic means water loving. They possess groups such as hydroxyl, sulfhydryl, and amide group. Their examples are proline, serine, threonine, cysteine, glutamine, and asparagine. Polar amino acids with negative R group. The dicarboxylic mono acid, aspartic acid, and glutamic acids are included in this group, or we can say that these are the example of polar amino acid with negative R group. You can see here clear in the diagram that these are different types of amino acid which can be classified based on their polarity. Last one is polar amino acid with positive R group. The amino acid carry positive charge on the R group and are dibasic monocarboxylic acid. They are highly basic in nature. The three amino acid lysine, arginine, histidines are included in this group. What is peptide bond? A bond called peptide bond link amino acid in a protein molecule to each other. The peptide bond is formed between an amino group of one amino acid and carboxyl group of another amino acid. This is the dehydration or condensation reaction in which one water molecule is formed. For example, this is the one amino acid in which the carboxyl group and this is the another amino acid in which the amino group, in which hydroxyl group from the carboxyl and hydrogen group from the amino group. They are combined to form the water and a bond is produced between the two amino acid, and this bond which is shown here in blue color, they are known as a peptide bond. A chain containing 3 amino acid and 2 peptide bond is known as dipeptide chain. And a chain with 4 amino acid and 3 peptide bond is called tripeptide, and a chain with many peptide bond is called polypeptide. And polypeptide chain of amino acid is simply known as protein. So most protein molecules are usually formed of 2 or many polypeptide chain. For example, hemoglobin, which is composed of 4 polypeptide chain, and insulin, which is composed of 2 polypeptide chain. Hemoglobin versus insulin. Hemoglobin is an oxygen-carrying protein in the red blood cell, which consists of 4 polypeptide chain, while an insulin molecule is consists of 2 polypeptide chain. The number in sequence of amino acid in a protein molecule is highly specific for its normal function. We can say that the arrangement as well as the number of amino acid in a protein molecule is very specific for their normal function. If 1 out of 574 amino acid in hemoglobin molecule is not present in specific position, for example, glutamic acid, if in 574 amino acid, if this glutamic acid is replaced by valine amino acid, then the normal shape of hemoglobin, that is globular shape, is changed into sickle shaped. Then the sickle shaped hemoglobin does not perform their normal function. So such type of hemoglobin cannot perform its function, and the person with sickle cell hemoglobin die. The disease is simply known as sickle cell anemia. The level of structure or organization of a protein. The protein can be divided into four different types based on their organization or level of structure. So primary, secondary, tertiary as well as quaternary structures are present within the protein. So depends upon the spatial arrangement of polypeptide chain. The protein can be divided into four types: primary structure, secondary, tertiary, and quaternary structure. The primary structure of protein. A polypeptide chain having a linear sequence of amino acid is called primary structure of a protein. So the sequence of amino acid in a polypeptide chain is called a primary structure. Amino acids are linked with one another through peptide bond. So these are various types of amino acid. When they are combined with one another through peptide bond formation, then the structure is this linear structure of amino acid is known as primary structure of the protein. When the peptide chains may acquire spiral shape, or may be present in zigzag manner. Or we can say when a polypeptide chain of amino acid becomes spirally coiled, then the structure is known as secondary structure of a protein. This coiling of peptide chain is called the secondary structure of a protein. It is due to the hydrogen bonding. So these are the clear diagram of secondary structure of a protein. Tertiary structure. When twisting or folding a polypeptide chain takes place, then the structure of protein is known as a tertiary structure. Quaternary mean four. It is the arrangement of multiple folded protein or chiral protein molecule and multi-subunit. Simply we call the quaternary structure of protein is complex structure of a protein. A variety of bonding interaction including hydrogen bonding, salt bridges, disulfide bonds hold the various chains into a particular geometry. So these are the quaternary structure of a protein. These are the primary structure, these are the secondary structure, tertiary structure, and quaternary structure of a protein. Properties of protein. You know that proteins are found in all living organisms involved in processes such as digestion of food, cell structure, catalysis, movement, energy, manipulation, etc. Protein are complex molecule, polymers of amino acid, long chain of amino acids are called polypeptide. Why are protein important to us? Protein make up about 15% of mass of average person. Enzyme act as a biological catalyst, which are protein. Protein performs storage as well as transport, for example, hemoglobin. It performs defense function, their example is antibodies. Proteins are hormone and their example is insulin. Ligaments and artery mainly forms by elastin protein. Muscle protein and the muscle respond to nerve impulses by changing the packaging of their molecule, actin and myosin. Here, nail and skin. Protein keratin is main component. The protein can be classified based on their structure into three types: fibrous protein, globular protein, and intermediate protein. On the basis of their composition, it can be divided into simple protein and conjugated protein. Based on their function, structural protein, their example is enzyme, hormone, pigment, which transport protein, contractile protein, storage protein, their example is a toxins. So classification of protein. Simple protein, those which give amino acids only upon hydrolysis, is known as simple protein. And conjugated proteins, those which give an amino acid in a non-protein groups upon hydrolysis, is known as conjugated protein. And those which are derived from simple and conjugated protein is known as a derived protein. These are the example of simple protein. Simple protein, these are globular protein, for example, albumin, they are easily insoluble. They are insoluble in water in dilute salt solution, coagulated by heat and found in plant and animal tissue. Simple protein globulins. They are sparingly soluble in water and neutral solution, coagulated by heat and distributed in both plants and animal tissues. These are the example of simple protein globulin, which is present in wheat and rice. Conjugated protein, as the name indicates, that protein combined with other biological molecule, which is known as a conjugated protein. Their example is nucleoprotein. A combination of nucleic acid with protein is known as nucleoprotein, which are present in the cell as well as in the virus. The protein can be classified into two types based on their shape: globular protein and fibrous protein. So fibrous protein, these are those protein which are fiber shaped, long thread like molecule. Their helical strands mainly form fibers. These proteins are insoluble in water, form structures of this tissue present where support is required. Their example is collagen, elastin and keratin. Globular protein, as the name indicates that they have globular shape, spherical in shape or ovoid in shape. Enzymes are mostly globular in shape, subdivided in two types of protein: albumin water soluble and globulin soluble in dilute salt solution. Classification of protein based on their function. Catalytic protein. All enzymes are protein and they perform the function of catalytics. For example, alkaline phosphatase, alanine transaminase, regulatory or hormonal protein. Many protein in peptide act as a hormone. For example, insulin, growth hormone. Structural protein. They form the structure. For example, collagen and elastin structure. Transport protein. They transport various types of substances. For example, hemoglobin carry oxygen. So form the function of defense, which is known as immune protein. Their example is immunoglobulin G, A, M, D, and E. Contractile protein help in the process of contraction and relaxation of muscle, and their example is actin and myosin. And genetic protein, as the name indicates, that the protein present in combination with nucleic acid. Their example is histone protein in the chromosome. Storage protein. To store protein for nutritional purposes, their example is casein in milk and gliadin in a wheat. So protein play an important role in formation of protoplasm. These are, we can say that the function of protein. The nucleoproteins are complex protein and act as the carrier of heredity material from one generation to another. All the enzymes are protein and they perform the function to speed up chemical by reaction without being used in the process. Transport oxygen to all cells of our body. Tanning of hides is actually precipitation of protein by tannic acids. Gelatin is obtained by heating bones skinned and tended in water. It is used in bakery goods. Importance of protein. Casein is another protein used in manufacture of button and buckles. Protein obtained from soya beans are used for manufacture of